Okay, so we're going to pick back up with talking about osmosis, which is going to be diffusion of a solvent. And most commonly, when we talk about osmosis, we're referring to water. In realizing that concentrations of water and concentrations of solutes are, related to each other, meaning that if solute concentration increases in a certain area, then water concentration is going to decrease. So they have this inverse relationship because every molecule of solute is going to kick out a molecule of water. So let's look at a couple examples of this. And so what we have here is basically, in this first scenario, we're looking at two different containers here that are linked together with a membrane between the two that is permeable to both solutes and water. So we have free movement going from side to side. And so what we see in this first part here, if I kind of reproduce that in this area here, and we've got kind of our freely permeable membrane. On the left side, we've got a certain amount of solutes. On the right side, we have many more solutes. You can see on the right side there. And I just kind of walk through this step by step because this tends to trip a lot of students up. So on the right side, we have increased solutes, and I'm just going to use S just to save the time from having to write it out. And we know that every molecule of solute is going to kick out a molecule of water. And so on this side, we're going to have decreased water content. Okay, so as compared to the right side, we actually have decreased solute concentration. So decreased osmolarity on that side. And then we're going to have increased water content as a result. Okay, so if we look at these two independently, we have free movement from side to side of both. What direction are the solutes going to move in? The solutes are going to want to move from their area of high concentration to their area of low concentration. So the solutes are going to want to move from right to left because they want to go from their high concentration to their low concentration. Now water is also going to want to move down its concentration gradient. So water is going to want to move from its area of high concentration to its area of lower concentration. So here in this area here on the left hand side, we've got a higher concentration of water, and it's going to want to move to the right side to the area of lower concentration of water. So we basically have this opposition or opposite flow, and because that membrane is permeable to both, if given enough time, both the water and the solutes are going to reach equilibrium where they're basically going to even out, and we're going to have the same amount of solutes and water on both sides of that membrane. So both sides are going to have the same osmolarity because the amount of solute concentration is going to reach equilibrium, and the amount of water is going to reach equilibrium as well. Let's look at the second example here. Here we have a similar setup, but what we've done is we've said, okay, our membrane that's separating the two compartments is only permeable to water. So it's not permeable to the solutes. So only water is going to be able to move. So what ends up happening is on the right side here, we have increased solutes and decreased water. On the left side here, we have decreased solutes and we have increased water. Okay, so our solutes actually can't move because this membrane here is impermeable to the solutes. You're given that information here. Okay, so water can move. So water is going to want to move down its concentration gradient. So as a result, water is going to want to move from the left side into the right side. And they're showing you that here with this arrow here. So what's going to end up happening is water is going to leave the left side of the compartment, and then it's actually going to flow into the right side of the compartment. And if given enough time, both sides of the compartment are going to end up having equal or identical osmolarity in terms of the concentration of solutes that you see in each. Now, you're going to see the volume of the solution on the right side. See, we've got much more solution on this right side than we have on this side, and that's because water was able to move across and able to fill up the right side of this compartment in order to create that equal osmolarity. Okay, so what ends up happening with most examples of our plasma membrane in our body is that there are a lot of solutes that cannot diffuse freely through the plasma membrane. So when we look at cells, a lot of times what we have happen is this scenario here, where our plasma membrane is selectively permeable and water is going to have access to flow in and out, but a lot of solutes won't. So solutes of varying concentration are going to result in altering the concentration of water on both sides of the plasma membrane, whether it's going to be, inside the cell or outside the cell. And it's going to end up affecting the cell by allowing the cell to either gain water or lose water as a result. Okay? And so this is what we talk about when we talk about the topic of tonicity. And this is the ability of a solution to change the shape of a cell by altering or changing the cell's water volume inside the cell. Okay, so how much water is present inside the cell? Well, we can do that by altering tonicity. And so what we look at here is cells that are in a hypotonic solution, cells that are in a hypertonic solution, and then cells that are in an isotonic solution. So one of the things to remember is that this is hypotonic solution. So we're talking about the fluid that cell is sitting in. So it's basically the extracellular fluid. And if it's hypotonic, hypo means low. So anytime you see that, it's going to mean low. So the solution around the cell is going to contain less solutes than the cells contain. Hypotonic, it means low. Hypertonic is exactly the opposite. So that means the solution around the cell is going to contain more solutes than the cell contains. And then iso means the same. So in an isotonic solution, the solution around the cell and the cell itself are going to contain the same concentration of solutes. So we have no net change in that case. So let's look at this a little bit. Let's do our hypotonic first because that was our first example on the other side. So what happens in a hypotonic solution? So we've got our cell, hypotonic solution. So that's related to the concentration of solutes in the solution around the cell. So I always draw, here's my cell, this would be inside the cell, and then this would be outside the cell. So hypotonic. So inside the cell, I have lots of solutes. Hypotonic means that outside the cell, I have many fewer solutes. So plasma membranes are semi-permeable, and those solutes can't travel through the plasma membrane, but water can. And so that's what we're affecting. We're affecting the water content inside the cell. So water is going to flow from its area of higher concentration to its area of lower concentration. So at this point outside the cell, we have decreased solutes and we have increased water content as compared to inside the cell. Inside the cell, we have exactly the opposite. We have increased solutes and decreased water. So in this scenario, water from the extracellular solution there is going to flow into the cell. The cells are going to basically become bloated and eventually burst, and we call that lyse, L-Y-S-E. Okay, that's what happens when you place cells in hypotonic solutions. Now on the flip side of that, we have hypertonic solutions. And again, you have to remember that the hyper refers to the concentration of solutes in the solution. So in this case, we're going to have lots of solutes outside the cell. Okay, and then very few solutes inside the cell in comparison. So outside the cell, we've got increased solutes, and as a result, because of their inverse relationship, we've got decreased water. Okay, we have exactly the opposite scenario that's going on inside the cell. So inside the cell, we've got decreased solutes and increased water. Water is able to cross this semi-permeable membrane, and it's going to flow from its area of high concentration to its area of low. So as a result, water is going to leave the cell. The cell is ultimately going to shrink. Okay, so it's going to go from normal size cell. This is showing you water leaving the cell. That cell is going to shrivel up and shrink because of loss of water. Okay, and I spend a lot of time going through this because I feel like a lot of students get kind of tangled up with this. So I try to explain it kind of step by step. Isotonic solutions, iso means the same. So it means that you have the same water and solute concentration both inside and outside the cell. So there's really going to be no net movement of solutes or water. It's gonna end up reaching equilibrium. Okay, so that's what we see in isotonic. Okay, so that's all of our passive transport. So now when we talk about active transport, what we mean when we say active is that we require energy to move a solute across the plasma membrane. And there's a couple different reasons why this would happen. So number one, if we're moving a solute against its concentration gradient, which means think about it as uphill movement. So if we're moving a solute from an area of low concentration to an area of high concentration, and we're going uphill, that requires the use of energy. It takes energy to be able to do that. Okay? Also, sometimes we have solutes that are too big to pass through those protein channels in the membranes, and in those cases we need to have active membrane transport like endocytosis, and we'll talk about that. And then in some cases the solute is not lipid soluble, which means that it can't travel through the phospholipid bilayer by itself, and so it needs a little bit of help. So there's two types of active transport. One of them, we've got active transport, which is going to be divided into primary active transport and then secondary active transport. And then we have what's called vesicular transport. And vesicles are actually just like little packages that contain substances that we either want to bring into the cell, and in that case that would be endocytosis, or sometimes we want to eject those vesicles from the cell, like it's something that we want to get rid of or we want to secrete, like a hormone or a neurotransmitter, and we call that exocytosis, so it's exiting the cell. So during active transport, one of the things to keep in mind is that the transport proteins, and we know those transport proteins are embedded in that plasma membrane, they are able to bind specifically and reversibly with the transported substances. So that means that this is a specific type of transport, and that once the protein binds to that particular substance, it's able to undergo a conformational change, and then it can reverse that change to get ready to bind to the next substance. Transport proteins are commonly referred to as pumps, and probably the the most notorious primary active transport pump is the sodium-potassium pump. And so what we have here is we have ATP hydrolyzing to ADP and inorganic phosphate to basically prime our pump so that we are able to move 3 sodiums outside the cell, and we're able to pump 2 potassium ions to the inside of the cell. And so this is what your sodium-potassium pump looks like. Now, there are some cases where we have primary active transport, most notably the sodium-potassium pump, which creates a concentration gradient that allows another type of transport process to run off of. Okay? And so if you look at this, here is our example of our sodium-potassium pump. And what's happening is it's constantly ejecting sodium to the outside of the cell. This is our extracellular fluid. So we end up building up an increased concentration of sodium on the outside of the cell. Now, in secondary active transport, also called cotransport, we are able to use a transmembrane protein, which looks like basically these, okay? And we are going to pair downhill movement, which means going from an area of high concentration to low concentration. So we're pairing downhill movement of one solute with uphill movement of another. Okay? So basically, if one solute is moving down its concentration gradient in a direction, it's able to actually pull another solute with it and move that solute against its concentration gradient. Okay? So it is secondary active transport. It does require ATP because we need to have this happen first. You have to have primary active transport happen first, and that requires ATP. And anything that requires ATP is considered active transport. Okay, so let's look at this. We have, through our sodium-potassium pump, we have ejected all these ions of sodium to the outside of the cell. That increases sodium concentration outside the cell. Sodium, however, wants to flow into the cell down its concentration gradient. And when we have secondary active transport, we are able to pull other ions or other solutes, like for instance glucose, in with the sodium ion, so that glucose is able to move against its concentration gradient into the cell paired with sodium. Okay? So, in this case, we have a symport system, which means that the solutes are moving in the same direction. So, sodium is coming into the cell down its concentration gradient. Glucose is also coming into the cell, but it's actually against its concentration gradient. So glucose is actually in higher concentration in the cell in this scenario, and it actually wants to go out. That would be going down its concentration gradient. But in this case, it's being paired with sodium entry into the cell, and therefore is called a symport system because they're both moving in the same direction. Now, there's some cases where we have what's called an antiport system, where we have solutes that move in opposite directions. So there would be examples of active transport where sodium is coming into the cell in that direction, but we would have a different solute that through secondary active transport is actually moving in the opposite direction. So if they're moving in opposite directions, that would be an antiport system. Okay, vesicular transport. So vesicular transport is basically based on using vesicles, which are these membranous sacs which contain substances. So we are able to take large particles across the plasma membrane. We can either bring it into the cell, which we call that endocytosis, or we can eject it from the cell, and that's called exocytosis. As you can imagine, having your plasma membrane, kind of invaginate and basically like engulf either bacteria or something else it's going to take energy, right, for that process to happen. That's pretty complicated. So vesicular transport is considered a form of active transport, and it's either going to use ATP or GTP as its energy source. Okay, there's 3 types of endocytosis, and this again is bringing substances into the cell. It typically is larger substances, and the way that we would engulf large substances would be through phagocytosis. Okay, and so you're basically going to have, maybe a pathogen or microorganism or bacteria that the cell wants to engulf. Maybe it's a white blood cell, and it's going to engulf that bacteria. So it's going to basically engulf it into a phagosome and then transport it through the cell. Maybe it's going to eject it through the other side. Maybe it's going to digest it within the cell through enzymes. But it's basically going to engulf that large microorganism within the vesicle, and then it's going to end up doing something with it. So phagocytosis is active transport where the cell engulfs a large particle. This is being used by macrophages or phagocytes throughout the body. This is specific because most of these vesicles do have receptors on them for various types of microorganisms or particles that they're engulfing. The second type is going to be pinocytosis. So in pinocytosis, what we have is basically the cell taking, I always like to describe it as like a little sip of extracellular fluid. So it basically is a little sip of fluid, and that extracellular fluid also includes solutes that are dissolved in the extracellular fluid. This is the process that is non-specific because there are no receptors in the vesicles. So this would be considered a non-specific type of endocytosis. And then our third type of endocytosis is receptor-mediated endocytosis. And so what we have here are specific extracellular substances. So some examples of this would be hormones, some hormones throughout the body. Insulin would be a specific hormone that would react this way, cholesterol. And so these specific chemicals or ligands are going to come in, and they are going to bind specifically to these protein receptors that are part of the vesicle. And then the cell is going to end up kind of pinching off that area, and that vesicle is now going to be kind of engulfed into the cell to maybe be released in the cell and then have an effect or be ejected the other side, or maybe lysosomes within the cell are going to digest that particular hormone or contents of the vesicle. So that is receptor mediated. So as you can imagine, it's called receptor mediated. This is going to be a more specific type of endocytosis. Okay, and then on the other side of things, we have the other half of vesicular transport. This is called exocytosis. So exo we can think of as exiting, and so this is basically the way cells are able to eject substances from inside the cell to the outside of the cell. So here we have another example of a vesicle, and we would call this a secretory vesicle because we're getting ready to secrete those substances to the outside of the cell, and it basically fuses with the plasma membrane of the cell. So the vesicle here is going to end up fusing with that plasma membrane, so it can now release its contents to the extracellular fluid. So there's a couple processes where we see this happen, and one of them is hormone secretion. So when we have endocrine glands that are secreting their hormones, most of the time they're going to do this through exocytosis. Also neurotransmitter release, secretion of mucus, and then waste secretion throughout the body. Okay, so back in the beginning, we talked about three main components that cells have. If you think about that for a second, we talked about plasma membrane. We've done a lot of talking about plasma membrane and how it's arranged and how we transport substances across that plasma membrane. The second part was the cytoplasm. And cytoplasm is specifically going to be the cellular membrane or a cellular material that is inside the plasma membrane. It's contained within it. So as part of the cytoplasm, we have the cytosol, which is going to be the fluid part. It's mainly water, also some other solutes contained within it. Cytoplasm also contains organelles. And so these are your mitochondria and your Golgi tendon organ or Golgi apparatus. These are the metabolic machinery of the cell, and they're the ones who do the work. And then we have inclusions, which are chemical substances that can be present inside the cell. And so sometimes these are like pigments. So some cells have pigment associated with them. And so that would be an example of an inclusion. So I have included the organelles, kind of big picture organelles. And so this is something that you would be responsible for knowing them to the level of detail that we've specified here. And the last thing that I want to touch base with here is the third part of all human cells, and that's the nucleus, which is the control center of the cell. This is where the genetic material is contained. Most cells in our body have one nucleus, although there are examples of some cells that are multinucleated, like skeletal muscles are huge, and embryonically they were comprised of many, many smaller cells that are fused together, and so they're multinucleated. There are other examples of mature cells that actually have no nucleus. So red blood cells have no nucleus, and that's why they have a lifespan of like 90 days, and they don't reproduce. And so once they die, that's it, and we have to make new ones. Nucleus is going to contain nucleoli, which is responsible for the manufacturing of ribosomes, and then the nucleus is also responsible for containing chromatin, which is basically the threads of DNA that allow us to form chromosomes during replication. So